So, first of all, can I just say what a pleasure it is to be invited by the Student Council to do something. Of course, you know, the future of all of our research and the future of ISCB depends on you guys. And so to be asked to speak is really an honor. So thank you very much. And I always struggle about what do I say? You know, what would be useful for you guys to hear? And I'm really not sure. So I've tried to sort of put together a bit of a mishmash of what was important to me and trying to interweave the science and the personal career business. So just to say, you know, I've been in this game now for over 50 years. And one of the few pleasures of getting old is that you can look back and reflect on how you thought when you were a student or a postdoc and how things have changed. And for me, I feel I was just at the right place at the right time, because these last 50 years have seen such a revolution in the world of molecular biology. Obviously, the DNA sequencing, the imaging methods, the computational power, and the AI methods have really changed the way that we work. And the impact on the life sciences is already huge. So if we think back, I suspect, you know, the word bioinformatics didn't appear until 1993. So I guess, would you put your hand up if you weren't born in 1993? Were you all, yeah, there are quite a lot of you who weren't even born. So this is quite surprising to me that these things have carried on like this. So the whole idea of using the data, of course, it's not a new idea. In physics, in all the other sciences, one uses the data that you have as the basis for hypotheses. And that's what really bioinformatics, in my mind, is about, bringing the data together to discover new things. And of course, the advantage of biology, which I still find after all these years amazing, is that all living creatures, plants, etc., are all built from DNA. It's just how we use that DNA and how it changes over time. And this gives a huge advantage actually to AI, because so much of what we see is inherited from the past. This isn't true if you think about basic physics. It's true in astronomy when you're looking at the evolution of galaxies, but it's not true in Newton's laws. They're the same yesterday, today, tomorrow. And so I think AI will be particularly powerful in the life sciences. And of course, we want to go, my personal interest has been in proteins and the details of protein structures, but we really want to take this from our knowledge of the molecules, the mechanisms that Melika just spoke about. We want to move that into the cells, the embryos, the whole organisms, and understand human development and aging and disease. But of course, the other thing that bioinformatics is really built to do is to be able to bridge biological disciplines. If we think about our biology, oh, I don't seem to have a pointer. Oh, can I have a pointer from somewhere? Or not, sorry.
Yeah, you cannot see from here, but it's like pointing on the slides there.
Ah, well, that's a bit difficult.
Yeah, so
all right, I'll do without a pointer. Yes, but I see.
Yeah, you have.
Oh, I see. Yeah. So we have all of these different sort of aspects of biology. And the only way that we can bridge them, and in fact, the multi-omics that we've just been hearing about, is another sort of bridging. And it's really only the computational methods that will allow us to do that bridging and to make the models that are predictive. And so to me, really bioinformatics absolutely is at the heart of the biology today and will be even more so in the future. So just for a brief interlude about me, I started out as a physicist and did my PhD looking at computational and biophysical studies of nicotinamide adenine dinucleotide. So when I started, I knew nothing, literally nothing about biology. And so for me, the last 50 years been a process of gradually learning about different aspects of biology, which is totally fascinating. But it is different when you've done different basic degrees, how you gradually begin to understand the field that you're moving into. And for my professional journal, I started doing my PhD in London. I went to Oxford for a postdoc. I moved back to London to first Birkbeck College and then UCL as a postdoc and then lecturer and professor, and then up to Cambridge to direct the European Bioinformatics Institute. So I've not really moved very far in my academic life, but that's partly because my personal circumstances have been such that I have been more or less fixed in one place. So, but happily, in this area of the UK, there are lots of different places one can work and lots of different opportunities. So my career, I started really, if I take it from the postdoc end of things, I really got into protein structures when I moved to Oxford, which was an experimental. I should say for my PhD, I did experimental and computational things. And in Oxford, again, I did both experimental and computational things. But then I had 2 children, and decided I was very fortunate. I was able to work part-time just for 2 days a week when the children were little. And I commuted actually to Oxford. And at that point, I decided it really wasn't practical to do experiments on a 2-day-a-week basis. I really would have had to have been in the lab a lot. But computing is different. And actually, it's very suitable for people who want to be a bit more flexible and have, especially those who are constrained to working in one area, perhaps. In the 80s, I went to Birkbeck and then increased to 3 days a week. So I worked part-time at that stage for 13 years while my children grew from 0 up to secondary school. I was in Birkbeck for about 10 years. Then I moved to UCL, which was all of 50 meters away, but it gave me kind of more independence. And actually it was, so I had my own group at Birkbeck, but in UCL, I was leading a small unit of both the experimental and the computational. So I had the opportunity, if you like, to perhaps organize things. And I don't know whether that was a good idea or not. My ex-supervisor, David Phillips, said, oh, you don't want to do any of this organizational things, just concentrate on your science. And I thought, but actually in this world, you can't do, because if you want to get grants, you have to do the organization as well as understanding the science to some extent at least. So I thought that was, he could just write, in those days, he would write a letter to the granting agencies and say, can I have 100,000 for this? And they'd give it to him. But I mean, now, of course, it's not like that. And for me, it was very much not like that. It was a case of really building a group of people together who would address some of the challenges. And then in 2000, I was approached to see if I would take on the EBI. And this was a big decision for me. because I was very happy at UCL, had a great group of colleagues. But I really feel, and still do feel, that handling the data and making it accessible is really, really important and fundamental. So I had about 3 thoughts. One was that, well, if I didn't do it and somebody else did it and made a mess of it, I'd be really cross. Secondly, do I know any other, and I would put in brackets here, male actually, who would refuse that opportunity? Because now it seems obvious. It wasn't then to me. And I thought, no. And so I thought, well, go. And I went on a secondment for 5 years just to see if I could cope with it. And of course, once you make that move, it was hard at the beginning, but you realize how important it, or I felt it was an important job to do. And I actually formally retired about 2 years ago, but I'm still busy and doing things I enjoy doing. So in terms of the structural bioinformatics, which I know most of you here probably, so is there anybody here working in structural bioinformatics? 1, 2, oh, a few people, good to see. Because for many years, I mean, that's one of the really good things about AlphaFold. It's really rejuvenated the field of structural biology, in my opinion. But when I started in the 70s, we had 20 protein structures, 20. So one could go and look at them in detail. And it was that detailed work. It was very different from the science of transcriptomics and metabolomics. And you had real hard data that wasn't, that you could validate. And we spent those first 10 years looking at all the different aspects of protein structures that are well known, but we looked at them because gradually we started with 20 and we had about 100 by the end of the 80s. So we had a lot of structures, and one quickly began to realize that we needed effectively a database, because you couldn't handle all of these structures and their names. And the worst thing was knowing which structures were homologs, because they didn't necessarily have the same names. And also that proteins that have the same names, like cytochrome C and cytochrome B5, were not homologs at all. They look completely different. So these things were as a physicist, they were quite amazing to me, that there was this inconsistency and this need to really understand this. And then as we got more and more structures, so of course our goal was always trying to predict, but we were sort of slicing and dicing the problem. So we try and predict where the helices were, where the beta strands were, where the turns were, all of these things. But then as we got more structures, our focus moved to the fold space, to looking at the whole structures, not at the little bits of the structures. And that was when, in fact, when we were starting to think about cath, I wanted a system a bit like the enzyme nomenclature, so we could have numbers to define the different types of fold from a purely practical basis, that otherwise we'd mix up the homologs and everything, so we had to do it. So that led us to develop the fold classification alongside Cyrus and Mertzin did the SCOP classification. We also discovered, so most of the crystallographers were hardcore physicists, and so they were very careful with their data. But as it got easier, people began to take shortcuts and they made mistakes. And it turned out that some of the structures in the PDB were actually wrong. Now this was, so it was quite shocking actually that this could happen, because it should never happen in crystallography. And so we developed these tools, ProCheck, which checks the structural geometry, the stereochemistry. And it showed that there was this beautiful correlations between various parameters and the quality of the protein structure, the resolution. And we then moved on at the end of the, well, really in the early 90s, to think about functions, because as a physicist, I must confess, for the first 20 years of my life, I really wasn't very interested in protein functions. It was their structures that really captured my, and I look back now and I think, how could I be so stupid? But I was. And I think we all have our different views of the science. But in the 90s, actually coinciding when I moved to UCL, I really made a very conscious decision that we would begin to look at functions and interactions. So we did a whole slew of analyses on protein-protein interactions, protein-DNA interactions, protein-carbohydrate interactions. There was so much to learn because they were all new structures and we could just look at them and try to understand them. And doing this is very different from an AI where you cannot look at cause and effect. We were trying to really look at that cause, the causal part of it. And then in 2000, when I moved to EBI, I carried on with structural stuff, in particular on the enzymes that we're still doing, never ending. So whilst I was at EBI, I've really done a lot on enzyme catalysis, metabolism, but also starting looking at aging. And I'll mention that a bit later. And in fact, we used neural networks way back in the 1990s, early 1990s, for beta term prediction. And they were more powerful than the standard methods. But it wasn't the deep learning methods, machine learning methods that are available now. But I wanted to predict protein structures, but I didn't have any good ideas about how to. I'd done all the analyses, if you like, but I didn't have, many students came and said, oh, I want to work with you on protein structure prediction. So I'd say to them, well, yeah, what are you going to do? What's new that's not been done before? Because many, many people worked on it. And it was really quite clear to me that it would be a technical solution and not one where I could contribute or indeed particularly enjoyed. I loved doing the analyses and looking at it, but I'm not an expert AI person at all. So when I moved to EBI, it was 50, the deal was I could spend 50% of my time directing the institute and 50% of my time leading the research group. Time management is the biggest challenge you will find. Even when I was a PhD student, actually, I wasn't very good at time management. It gets more difficult. Let me just warn you of that. And whilst I was at the EBI, our approach or our understanding about perhaps our ambition to go from individual molecules to whole systems really grew. So when I joined EBI, there were just 5 databases. Now there are well over 40 databases. And so over that time, as I say, our ambition to really understand the systems part, this bit down here, to go all the way from the genome right down or up to the whole human organism, or other organisms indeed. And one of the joys of EBI, of course, is that almost all our data is completely open. And so people often think of well, I've heard the EBI called the Data Liberation Army, because people in general there are very much in favor of making the data open. Of course, it's not their data, I should say. This data comes from all around the world. All these databases, every single one of them is collaborative. So they're done on an international basis. And this is the only way to solve some of the problems that we heard about from the panel by international agreement. Just to give you a scale, I can remember when I went to EBI, we had 100 terabytes of data. We now have 309, this was last year actually, petabytes. Every day there are over 100 million hits on the EBI website, which is huge. And I think it speaks volumes to the power and the importance of sharing data for everybody to use. And there are lots, I mean, there are 36 million unique IP addresses. Now, some of those, obviously, duplicate, it's always hard to measure. But the impact, I think, is really huge of doing the data sharing. But of course, one of the things when I joined EBI, I said, we're not just going to do molecules, we're going to look at medicine and agri-tech and do all of these other things. And people looked at me and smiled and, but nobody came to do these things. That has changed completely. We had our EBI research retreat 5 days ago, and every single project was in an area which was directly relevant to either health or, agriculture or infectious diseases or whatever. And so I think this has changed in the last 20 years quite a lot. So just to mention just some of these, the AlphaFold database, as you know, has completely changed the way. I think the important thing is that biologists, experimental biologists, can now be interested in a protein and they can get a picture of a structure. It might not be very reliable, but they're beginning to think in 3 dimensions. And that, to me, is really important. And this database that's set up with 200 million protein structures. I started with 20. I mean, this is phenomenal. And of course, the whole thing was based on the PDB and having access to those structures that were determined. In terms of these applications, of course, the obvious are the molecular medicine, agriculture, food, and environmental sciences. So if we just very briefly, 3 snapshots, way back actually in 1993, I worked on looking at mutations in P53, which, as you know, is involved in almost half of all known cancer. And of course they are at the point where the protein recognizes the DNA, that's the site. I said I moved on to aging, and I put this slide on before I knew that Melika was here. Melika was a PhD student in the lab, brilliant student actually. And for me, this learning about transcriptomes and trying to join up, so to identify drugs that produce the same expression changes as an expression profile of aging. I mean, it was preliminary, I would say. Maybe now one could do it again and see how it had changed and improved. But it was a real eye-opener for me, because it was looking at this data that is rather different from protein structure data. And I was educated by Melika in many ways. And we also looked on the agricultural side. We've been working on P450s and the enzymes. And, P450s are really important in the plant kingdom. And they are a family that has evolved and diversified so that it creates a huge, even though the structures, here, you can see, this is a superposition, sorry, I don't like the moving background, but anyway, many different structures that all look similar, but they actually work on their enzymes that work on very many different types of substrates, and they've evolved to be specific for different substrates. And these are critical in understanding things. So if we think about AI, obviously, in order for it to be really powerful, we need to ask the right questions and have the metrics to measure the outcome. That's really important. We need computable high-quality data. Obviously, the new methods in AI have really changed, and they are fantastic. And most of them, I don't understand the basis of them. But I do worry about people using them as entirely black boxes, I must confess, because I think you have to understand. I think that's what, frankly, DeepMind brought to the problem of protein structure prediction, to use the right methods to handle that data. We need a lot of compute. We need benchmarking continuously. And we need this community assessment of what the results are to try and compare and standardize all the problems that was described by the panel. We really need to address them. And I don't know if you can read this okay, but this problem with medical research and medical data is a huge problem, absolutely huge. And it can be solved by doing cohorts, like the Biobank, UK Biobank, which is incredibly powerful. But ideally, of course, we have data for almost all human beings around the globe. The problem is how to standardize it, how to use it, how to incorporate it. And so I would argue that, it's really taken 30 years to build up our ecosystem of molecular data resources, like a TBI or NCBI or whatever. For medical research, I think that process is just starting. It really is a change of culture. It's a change of scale, because the medical data is much larger than molecular data. The data are even more sliced because of things like the country or the policy or the politics. It's really hard to handle these data. And of course, there are a lot of potential financial gains associated with producing new drugs. And that brings with it a whole new slew of constraints that we really don't have with most of the molecular or the pathogenic. As soon as you come to human data, the privacy is absolutely tantamount, and the trust of the people whose data it is really important. And so I'm quite convinced. So in the UK, we have something called HDR UK, Health Data Research UK, which is trying to make access to data, clinical data, easier. There's a talk tomorrow in the UK track about some of that. And that, I think, is really important, but it's going to be hard. It's not going to happen overnight, I don't think. So one of the other things, and that also relates to the panel, was the building of a network across Europe to really collaborate on some of these data issues. And that network became Elixir. I was responsible, I have to say, in terms of time management. I think I could cope with the EBI in my research. Not well, but I did it. When Elixir came along as well, that became totally impossible. Trying to coordinate 20, 30 European countries to come together to build this network. And it was done with a group of like-minded souls, many of whom are related to ISCB here. And we built the Elixir infrastructure that is now led by Tim Hubbard. And they're working in different areas, looking at all the things that we all need to look at: compute, data, interoperability, tools, and training. I would say there, to me, it's not the most important part, but it's the most necessary part, is to have the data and the data organized. and countries remain somewhat reluctant in terms of sharing data, especially the medical data, and understandably so, but I think one has to try to overcome those barriers to deal with this better. So just to conclude then, these were some thoughts from, I had to give a talk about personal academic careers. And I had the pretty standard route: undergraduate student, postdoc, group leader, head of unit, head of institute, and then back to being a part-time researcher. And now I can add another dot there, back to being retired. So it's sort of, that's the core of it. But of course, one doesn't just do your academic job. There are lots of other things that come along with being an academic. And I was involved in spinning out a company. I didn't make any money, but the Kemble databases is a result of that company, and so I'm quite pleased actually that that happened. Scientific governance, I was on ERC Council. I'm on lots of boards. And I can remember when I was your age, I used to think, what are these people doing, fiddling around on these boards? But actually, they are quite important. And you realise perhaps a little more as you get a bit older how important it is, the governance of lots of different efforts. Really is important that it's done properly. I would also say that each stage along this route is very different. What you do on a regular day-to-day basis when you're a PhD student is different from what you do when you're a group leader, is different from what you do when you're head of institute. It's really an interesting thing, but I've put what goes round comes around. The people that you meet along this route are really important for your overall career. They come up again and again. You will find you will meet people. I meet people that I, knew as a postdoc or knew as when I was a new fellow and things like that. And it's really important to form those relationships and to form good collaborations. You don't have to work together, but having colleagues who trust you and who you trust them for advice is really important in that. There are transitions that you have to go through. As I said, it's different. So when you first arrive as a student, I went from a physics department to the National Institute of Medical Research. It was quite a shock to the system. I didn't know the language. I didn't understand what they were talking. So this was, for me, was a big transition. And actually, I nearly gave up halfway through to become a medical physicist in a hospital because I wasn't sure why I was doing what I was doing. And then they didn't give me the job. So I went back to being a PhD student, finished my PhD, and by that time I was kind of hooked. When you become a postdoc, you have that bit more independence, and it's important that you take that independence and work through. Sometimes it's not so easy if you're allowed to, but as long as your supervisor really supports, because the point of a postdoc is actually to learn and to move towards being independent, to do your own research. And in research, the most important thing is to ask the right questions, to ask the right questions that are feasible and timely. And that really you have to work on as a postdoc. When you have a group, again, it's different because you're not exactly a social worker, but there's an aspect of that in it because you have to, look after your students. And that's, for me, that was the bit that I always enjoyed most, actually. But it's quite interesting. When you become director of an institute, it's another transition that you have to make. And I can remember walking into the institute and the people there called me the boss. And I was horrified because I've never bossed anybody, and I didn't expect to boss them, but they expected the director to be in charge, which I guess I was. But I had to learn how to do that and to manage these expectations. And of course, funding and admin are really important, and you have to absorb that as you get more senior. So a few reflections. I have just loved being in research. I wouldn't change a minute of it. Partly because the field has been so exciting. It's not just, it's not just sort of crept forward. I mean, it's moved forward in leaps and bounds. It's great working with other people. You have to publish, and that's really important that you learn how to write papers when you're a student, because you will ultimately, I still feel it's your CV and your publications that do matter. I have to say, you have to kind of find a niche. For me, it was protein structures, because I love them. I mean, they're so beautiful. Why wouldn't you love them? But it's different for different people, and I understand that. Some people love the details of the algorithms, so you need to find your own niche in that. I have to say, you've got to work hard, and it doesn't get easier. It really, because you have to multitask when you're kind of in charge. I think you have to be optimistic and a little bit arrogant as well. In my observation, in order to really push a field forward, you have to believe that you can do it, that it's so you have to bring with it that certain arrogance that you can make a contribution, and that is quite important. And as I say, forming friendships and collaborations. For managing, I didn't have any training. I mean, I look back now and think, where angels fear to tread. I didn't really have the training. And I think as a research PI, you really are, you have to be in the weeds. You have to understand all the details as much as you possibly can. When you're running a large institute, you also need to learn how to delegate, to trust implicitly other people, and you have a lot less control over your time because you're always reacting to outside pressures. So you need to learn to delegate effectively and clearly, and that's a challenge for most researchers because most of us want to be in there and not sort of away from that. The other thing is that communication is vital. And as the Institute grew, I realized we had to change because word of mouth wasn't enough. We had to do town halls. We had to tell everybody because it's important with a group. You have your group meeting, you know everybody hopefully well, and you know what everybody's doing. When it gets larger, you can't operate on that basis. So leadership, you may think, oh, I'm not ready for leadership now. I'm in this. I'm at this stage of my career. But actually, at every stage, there are opportunities for leadership. And if you want to do it, some people do, some people don't want to do it. But if you do, then take it on. Don't step forward. And it is daunting, but you grow to learn how to handle it. It is a bit exhausting at times, but I very much enjoyed thinking about the strategy in the future. You can imagine a TBI. Well, which databases? What's going to happen? How do we manage it? Who do we interact with? How do we get the funding for it? There are all these questions that are outside the nitty-gritty research questions. And whilst those are important, and you have to do it, I think the model at EBI where you retain your research group, even though you're in charge, is a brilliant model, actually, because you very quickly learn that actually you can have hypotheses about management, but if you get down to the nitty-gritty of the data, like you can't combine the wretched data sets, you have to do bulk corrections, and even then it's not really very good, etc. So that's important. But I would say, when I was younger, I didn't really have that much interaction. I didn't really understand leadership in science. But I think, maybe I'm biased, but I think it is more important than I realised then, because the leader can really shift the culture in an organisation, in a group. Your own research group actually has a culture that you are responsible for and you should try and shape to be positive. So I think that's important. Oh, on being a woman and a mother in science, I've rarely been conscious of negative discrimination. Perhaps I've just had a fortunate route. Maybe coming from crystallography, where Dorothy Hodgkin was one of the first and had a Nobel Prize, changed the culture in that field. But I always felt very accepted, and sometimes actually, I have to say, I think there was positive discrimination rather than negative. As I say, I loved having this scientific group and an extended family, and having retired, because I don't have a group now, that is the thing that I miss, actually. Having new data to look at and new things to think about and what's going on. But it's okay. I've got lots of other things to do. I do think it's an option for being, for a working parent, male or female. And I think you don't have to be pushy, but you do have to be a bit ambitious and, look for opportunities and take them when they present themselves, because they'll be looking for things to do. So just to conclude, thanks to my family, thanks to all the members of my group, past, there aren't any present ones, but we had a wonderful day when we all got together. It was really lovely to see everybody and hear about all of their different journeys. And of course, thanks to everybody at EMBL EBI, UCL, Birkbeck and Oxford. And each of those places shapes the way that you think, and that is quite important. I would say that as a scientist, you do have to work hard. And is it possible to have a healthy work-life balance? Because I think quite a lot of scientists don't have a healthy. I've met people over my career who really have not. And so I think I'm not saying the way to do it, the way I did it was, at certain times, either home or work would tend to take precedence. When I was leaving the institute, I couldn't have done that when I had little kids. When I had little kids, you've no option. They have to be your priority, because you've just got to go and meet them from school at 5 o'clock. There's no option, or 4 o'clock, whatever it is. So it's not easy, but I do think it's possible and everything. So I think really the future is yours, certainly not mine. And I hope you have as much fun as I've had. Thank you.
Thank you so much for that beautiful and inspiring talk. I think your words really resonated with a lot of people in the audience, and they certainly did with me. We definitely have time for some questions from the audience. I saw a hand go straight up in the back there. I'll be with you now.
Thank you very much. It has been most inspirational, and I am very lucky because I am here listening to you and talking with everyone. But your talk was fantastic, really. I'm so happy. It's about this balance with your personal life and your work in the lab. You have talked about real truth. Always we have a lot of more time for the lab, and another times we have more time or more energy for the family, and the balance doesn't exist sometimes because you have to put effort sometimes more to your family because you have problems, or you have to change this, depend of the context, of course. And what do you think about this, the help of a good partner, to select good partner, because you have husband, you have children, but good partner is, I think, is essential, because I am recently I have been married. I have a husband in the science, or too, fortunately, because he can understand me. But despite this, what is the value, the importance to select a good partner to continue in this incredible, incredible, I don't know, in your life,
Yeah.
like women or men with family
Yeah,
love.
I think it's
Thank
the same. It doesn't matter whether you're a man or a woman. The way I think about it is that you have your personal life, and I've seen many examples. Well, obviously, we're all different. We all do things in the way that we do them. So for me, my home life and my work life are quite separate. My husband is not an academic. He worked for a computer company. And we just, partly because we've always lived in the same place, and I've basically commuted an hour's drive, either to Oxford, Cambridge, or London. It's an hour. And so just by the geometry of it, I kept my work life and my home life separate. But I've seen other examples where both partners are scientists, sometimes working in the same area, sometimes in different areas, and everybody manages it differently. I mean, I think the answer to this is to do things the way that suits you. Do what really suits you and suits your environment and your personal preferences. I don't think there is a right answer. It's just like there's not a right answer how you do science. There are many different ways of doing science. And so I think we all bring with us not only our scientific credentials, but our personal credentials. And if you just do what you think is the right thing to do, and it's not always easy. We know that. We all have to be tolerant and deal with things as they come up, and so you have to move with the wind and make things work. So I often think, I mean, it amazes me the way that science moves forward, actually, because if you think of the models that we have, there are students for 3 years. Those are just come in, they don't know very much, they're not an expert in the area, and yet these students produce amazing science. But then they might go off and go to work for a bank or something, dependent on what they want to do. And so somehow science progresses despite the challenges. And I often think in terms of achievements, you achieve despite all of those things that say this isn't going to work, whether it's a personal thing or a programming or access to a computer or whatever it is, just trying to find the right way. And I think you need tolerance. I suspect the key quality in a good partner is tolerance for when you're doing that. Having said that, my partner isn't particularly tolerant, but it would be nice if he were. So, you know, it just depends.
Hi, thank you. Working? Can you guys
There
hear?
you go.
Yeah, so I'm Hisham Zealand. I have a twofold question, but first, I'm really grateful when I was learning bioinformatics, ABI and EMBL really were fantastic, and I still remember when I start working, and then you show a PI at the time, like, you know, the protein structures and stuff, and they go like, where's the Western blot? And you go like, well, this is computational, it's not lab, but, you know, those sort of, we have come a long way since then. What I really found fascinating in your speech is your background and coming from across disciplines. And so that interdisciplinarity is really important, and it gets things done rather than somebody who's just immersed in one field. So my question to you, the first part of the question is, how do you know, or how did you know at the time that a particular question or pursuit was actually the right one? You mentioned in your speech here, you know, surrounding yourself with people with complementary skills, and knowing that you don't have all these complementary skills. How do you know, like, what gives, where do you get that intellectual equity, so to speak? And the second question is more reflective. You being in a field, how do you position everything in order to inform policy, coordinate all these governments, harmonize, you know, the data sharing agreements and everything? Yeah, so that's my question.
So how do you know it's the right question? Well, of course you don't. You don't know it's the right question. But for me, you know, scientists are also quite selfish. And I chose the questions that I was interested in. I mean, it was as simple as that. I wanted to know about protein structures in all their nitty-gritty, gruesome detail. And I was just fascinated to look at the results, because we, even in the old days, we had these new structures, and we all, you know, a new structure would come out and we'd rush to the library to see what it looked like. We didn't know whether there'd be strands or sheets or, you know, whatever. So it was absolutely natural for me. It never felt, I never had to think about what I'd do next. It was just obvious to me what we should do next. I think then the second question was how to how to coordinate things, and the answer is, of course, with other people. Because when you're trying to work across different European nation states, each nation has its cohort of bioinformatics people. And each one is quite different from the others. And one of the things, I mean, it was fascinating doing Elixir, because we got to travel around to see all the different European nations. And they all operate really quite differently. And the same is true even more so in the medical field. That's another question. But the only way, so what we did, we said we will come and discuss with the bioinformatics people what Elixir might be for that country X. And then the local people would go away and discuss what they thought they could bring to the table, what they could use, and then together we would try and go to the ministry and say, give us some money to fund all of this. And so it was quite important, in my mind. One of the chief outcomes of Elixir is very much that the bioinformatics people in the country came together and had a discussion together and tried to work out national priorities. And those national priorities fed into the whole of Elixir. And I think it's, you can't, you can influence, but ultimately the countries decide for themselves what is their priority. And sometimes it depends on the agriculture or the fisheries, or, different countries are very different. And that's, in a way, it's the great pleasure of Europe, that we've got this variety of different environments that have evolved. And yet, in Elixir, we can work together, hopefully, to do that. Okay.
Hi, thank you so much for your this fantastic and inspiring talk. I just have a question. You mentioned, like, for example, the term bioinformatics started to appear in the literature around 1987, 1989. And then I just remember that about Margaret Dayhoff in 1965, when she published the first
that's right.
atlas of protein, sequence of proteins. So I wonder, because I'm passionate in the history of bioinformatics, between 1965 and 1987 that the term is coined, what happened in that gap during?
Well, so
Sorry, but maybe.
it's very interesting. So if you, what happened was that we in protein structures and structural biology were working through to understand the proteins, etc. In parallel, of course, there were the developments in molecular biology and cloning and the ability to sequence, because I think that probably is the most single important change that I didn't see coming. I mean, I knew the number of structures would go up exponentially. That was entirely predictable. But these new technologies really are game changers. And the people in the genomics world think that they discovered bioinformatics, you see. So it's quite interesting. And EBI is a very genome-focused place with the Sanger next door and, being the center of genome sequencing in a way in Cambridge. And the structural biologists were rather separate. And so it wasn't that nothing was happening. It's just that for so long, we as structural biologists were focused on, there's the generality of proteins, which is fascinating. The whole universe of protein structures. I mean, it's beautiful, and it's like discovering a new continent. But at the same time, the individual proteins are what really matter for specific functions, and the protein families. And so so much work goes on sequence analysis because it's in 1D and it's fast now. There was a part of the field just ignored the proteins. I mean, that's how it sometimes feels at EBI, I must confess. Even today, the focus is very much on the genomics. And so it wasn't that nothing happened, it was just that it was rather separate.
They were not connected at that time.
Well, of course they were. They were connected. But I have to say, when I joined EBI, one of my first requests was that it would be possible to go from a nucleotide sequence to a protein sequence, please, which to me seemed absolutely basic. But it's actually quite complicated because of making sure that all the discussions about the transcriptomics and making sure you've got the right metadata, that there were problems there. So I think that they evolved, the structural biology was mainly done by physicists, actually. The sequence data, I mean, the computing has developed such a long way. So when I first used PDB, so my job when I was a postdoc in Oxford was to receive the 12-inch magnetic tapes that had the PDB structures on them. So I would get them every 3 months from the PDB. We'd actually get the coordinates on these big tapes. And, that came in the post in a big package. And I was the person whose job it was to mount these on the system. And if you think, then when I moved to to UCL in the 1990s, we started to get email. Amazing. You know, I could send emails to people all around the world instantly. I mean, it was fantastic. So the communication immediately, almost overnight, changed. The other thing was the compute. When I was at Birkbeck, we had one Evans and Sutherland machine for the whole department. In fact, it wasn't just our department, it was King's and Imperial as well. And so we had to book time on these machines to look at the proteins. So I'd perhaps get an hour a week if I was lucky. When I went to UCL, for the first time, we had desktops. So everybody in my group, and they were cheap. I could afford, with my startup grant, which was minuscule, to buy a desktop for all the people in the group, and they could program all day. I mean, it was a revolution again. So these sort of technical developments, along with the sequencing, that opened up the world of life sciences, really. So it wasn't just one protein, one species, but all proteins, all species, all sequences. So these technological, and the transcriptomes, I mean, there was no transcriptome till the early 1990s. And so it's, you know, the game changers are the technologies without a doubt. And that's what's made computational biology so important, really. But it was obvious it was going to be, because there's no alternative. You know, the only way you can join up species is actually to compare sequences, which you have to do computationally. I mean, it's quite interesting looking at that side of it, the development. But I assure you, we weren't doing nothing. We were busy.
Thank you so much.
Thank you so much for the questions and great answers. I'm sorry, I would love to continue this all evening, but I think we're going to have to wrap up now. But Janet, am I right in that you'll be attending the main conference?
Yes, I am indeed about to go and do my bit. We're introducing
Fantastic.
John Jumper, so I encourage you to go and get to the lecture theatre early, because there isn't room for everybody. So you need to get in, otherwise you'll be in an overflow room. So I do encourage you to get there on time, at 6 o'clock. But I'm around for the next until Thursday, so please ask me questions. Especially, I've not talked at all about enzyme catalysis, which really is what I'm obsessed with. So if anybody's interested in that, do come and chat to me. Okay, thank you very much.
Well,
Good
thank
luck.
you so much. And we have a small cake and barricade.